So in the traditional plant breeding and animal breeding concepts, in the topic of hybridization, we are transferring the good genes along with the genes which we are not interested in, and this problem can easily be overcome with the help of genetic engineering, because the technique of genetic engineering includes creation of recombinant DNA, then use of gene cloning. Gene cloning means making copies of the gene which you are interested in, and then gene transfer. And this rDNA technology or genetic engineering or otherwise the molecular engineering technology has overcome this limitation, the limitation of transfer of undesirable genes of the hybridization concept, and allow us to isolate and introduce only one or a set of desirable genes without introducing the undesirable genes into the target organism. I hope this point is clear to you, that in this concept we are transferring the desired genes. Maybe we are interested in one gene or two or a set of genes, but along with that all undesirable genes also will go. Otherwise, if the genome it does not carry all the genes, then definitely the zygote cannot survive. But in the case of genetic engineering, we simply alter the genetic material and we transfer that into the host, and now the host will show a change in the phenotype. But in this concept, we are only transferring the desired gene or the set of desired genes. Undesirable genes are not transferred, and this became possible because of discovery of the vector. Vector means the carrier, we'll be learning about it, the plasmid, and then the two beautiful enzymes, cutting enzymes and joining enzymes, which are called as molecular scissors or restriction enzyme, DNA ligase or molecular glue. Now there is one more thing which you need to remember, that genetic engineering depends upon two main concepts. One is how to modify the genes, and second, how to maintain the conditions. That means the chemical engineering. So I'm talking about this again from the NCERT book itself. So the principles of biotechnology, there are two core techniques that enabled the birth of biotechnology or modern biotechnology. First is how to do the genetic engineering. Now what is genetic engineering? Techniques which are helpful to change the chemistry of the genetic material. Now remember, genetic material can be DNA or RNA, but of course in maximum organism you will find that the genetic material is DNA. And after changing the chemistry of the genetic material, to introduce these into the host organism, and then we will find a change in the phenotype of the host organism. Change in the phenotype means the features will change. That means if a gene of synthesizing a particular protein is there, then the host will start showing now a different protein. That's why it is called as phenotypic change. Now second thing is the chemical engineering, also called as the bioprocess engineering. Now how to maintain the desirable, sterile, bad microbe free environment, so that the environment around the host organism remains good and this host organism reproduces, because we all know the microorganism reproduce when they are facing the favorable condition, then only they perform asexual reproduction. So the microorganism or the host organism will reproduce asexually if they are feeding a lot. So how to maintain good environment around the host organism, so that the organism continues to feed, and then after feeding it will start reproduction, because more it reproduce, more this quality is passing on to the next generations. And imagine you start with one single E. coli or one single microorganism, and it starts reproducing asexually by binary fission, and after few generations there are so many microorganisms for you, and all of them are genetically modified host organism, and they all are living industry or machines for you for preparing product of your desire. That means This is the concept of chemical engineering, because we need to learn how to maintain sterile condition. What is sterile? Sterile means microbial contamination free of all bad microorganism, so that there will be growth of your desired microbe. That desired microbe is the host organism. That microbe, it could be even eukaryotic cells also, for example yeasts, to obtain the products in the large quantity. So two things are important in the modern biotechnology: how to change the DNA and how to maintain conditions, that is the chemical engineering. I hope this is clear to you. Now the next main point to understand is that I'm reading again from the NCERT, page 194. Do you know the likely fate of piece of DNA which is somehow transferred into the alien organism? Imagine I give you a piece of DNA and I ask you to introduce into the single cell organism, or maybe a single cell taken out from a multicellular organism. What do you think should be the fate of that cell? Of that DNA inserted inside? Definitely this DNA is not going to survive inside, because the cellular level of defense will start, and the cell is going to digest this alien piece of DNA. So most likely this piece of DNA will not be able to multiply, and it will not pass on to the next generations also. But imagine if this DNA can become a part of the genome. Then what will happen? Every time the genome or the genetic material of the host cell is dividing or replicating, or cell division is going on, then the alien piece also will successfully pass on to the next